Okay, so today our topic is the digestion of protein. In the previous video, we were done with the digestion of cellulose. So today we will be discussing about the digestion of protein in our body. Okay, first of all, we are eating a protein from in the form of food. Okay, so after that, in the buccal cavity, the digestion of protein will not occur because they don't the in buccal cavity we are not having a proper appropriate you can say that proteolytic enzyme, so that's why in buccal cavity the protein will not get digested. Okay, so from in the form of bolus from the buccal cavity the food will move into the esophagus, and with the help of the peristalsis movement it will move into the stomach. As you guys all know, there is no enzyme in the esophagus too. Only there is a mechanical and the chemical digestion. Okay, so it will occur in the esophagus, so they are not having any kind of appropriate enzyme. Okay, so if you're talking about the stomach, so as you guys all know, stomach is having we are having two proteolytic enzyme that can help in the digestion of the protein. Okay, but they both are in the inactive form. We have discussed in the stomach that we are having a one enzyme, that is pepsinogen, that is converted by the HCl, okay, which is a cofactor, and it was converted into the active form, that is pepsin, and it is a holo enzyme, as you guys all know, and this is an apo enzyme because this is inactive. Okay, so first one is this, and if you're talking about the pH of the pepsin, so that is 1.2 to 1.8. Okay, second proteolytic enzyme we are having, that is a prorenin. This is also in the inactive form. The pepsin will work on it and become a prorenin will become into the renin, which is the active form. Now they both will work on the protein because they are converted into the active form. Now if you're talking about the renin, so renin is mostly present in the children's, not in the adults. They are present in the infants. Okay, so but in our body the renin is there, but in the low concentration. But as you guys all know that for the milk for the milk protein, the in you can say that adults the pepsin work, but if you're talking about the children, so the renin will work. Okay, work on the that will work on the protein of the milk, which is called the casinogen. I'm talking about the infants. Okay, so this casinogen will convert into the casein by renin, sorry. Okay, then casein will convert into the paracasein with the help of the renin. Okay, this will react on the calcium ions. Okay, if it will not react on the calcium ions, so as you guys all know, we have discussed the coagulation process, so the coagulation will not occur if it will not react on the calcium ions. Okay, then this paracasein will convert into the calcium paracaseinate. Okay, these are what? These are the conversions with the help of the enzymes. Okay, now if you're talking about the calcium paracaseinate, so on that one the pepsin will work and it will convert into the peptones. Okay, this is what? This is the small chain of the protein. Okay, then as you guys all know, in stomach it's done. Now if you're talking about the duodenum, which is the first part of the small intestine, here as you guys all know, we were in duodenum, the secretion was bile secretion was coming, as well as the pancreatic juice was coming into the duodenum for the digestion. Okay, so as you guys all know, in bile there is no enzyme for the digestion of the protein. okay, but in bile there is just a emulsification occur, but otherwise they are not having any kind of enzyme. If you're talking about the pancreatic juice, we have discussed the endopeptidases, exopeptidases in the previous video of the pancreas. We have discussed all these all, okay. So if you're talking about the pancreatic juice, we were having a endopeptidases. So in that, if you're talking about, we were having a two enzyme that was in inactive form, that was trypsinogen and chymotrypsinogen, okay. This was converted into the trypsin with the help of enterokinase enzyme. This was released by the duodenum, as you guys all know, and this will convert into the chymotrypsin in the active form, which is active form, with the help of the trypsin. Now the trypsin is become into the active form here, so this will work as a autocatalyst. What does it mean? It means that that will activate all the inactive enzyme. Let me tell you how. This was the endopeptide. If you're talking about the exopeptidases, what is peptidases? Peptidases means that these are the enzyme which break the, you can say that the endoexopeptide, one will convert into the smaller chain, and one will be break out the terminals of the protein, okay, or you can say that amino acids, okay. So these are the enzymes that will work on the protein especially, okay. So if you're talking about the exopeptidases, so there we were having two enzymes was there. First one was the procarboxyl peptidases, and it will convert by the trypsin. As you guys all know, I said you guys it will work in the as the autocatalyst. So by the trypsin it will convert into the carbopeptidases, okay. So now the protein will convert into the small peptones with the help of a enzyme that is trypsin, as well as the chymotrypsin, which are now in the active form, chymotrypsin, okay. So next we are having a, you can say that these peptones are like small peptones are converted. Now these small peptones will break down. We have to do a digestion of the protein. So first of all we will be the breaking out the protein, then it will got digest. So small peptones now will break down uh, into the amino acid, little bit amino acid, with the help of the pancreatic juice and the intestinal juice, okay. So that on that the carbo oxypeptidase will work, which was in the active form, as well as the amino peptidase, okay. These both are what? These both are the exopeptidases, okay. Now if you're talking about this, so these are the exopeptidases, and which will break the terminal bond. Now we are having a amino acid, but in the dipeptide form. Dipeptide form. What is dipeptide? Which means we are having a two peptide bond, or you can say that two amino acid, two peptide bond, okay. It will break by the peptidases finally into the amino acid. Now the amino acid will got absorbed easily, okay, in the duodenum, or you can say that digest into the duodenum, or in which part? Small intestine, okay. So dipeptidases, it is an enzyme which is secreted by the duodenum, or you can say the pancreatic lining, and it is secreted by the enterocytes, which are absorptive cells, as you guys all know, okay. This will work when body need amino acid, okay. So here you can see the digestion is occurred. Now amino acids are found by breakdown of the, you can say that protein, by a different enzymes, okay, which were working on the protein. So now the amino acid is there. Now it is now digested into the duodenum. Okay, now you can say that the absorption of the protein will occur into the by the villi, as you guys all know, but more villi are in the ileum, so the absorption will occur in the ileum. If someone asks you that nucleic acid is break in where, so you can say that the nucleic acid break into the jejunum, which is the second part of the small intestine. And if someone asks you that nucleotide will break into the nucleoside, so in nucleoside we were having a pentose sugar with a nitrogen base, but in this we were having a pentose sugar as well as the you can say that phosphate group as well as the nitrogenous base. So it will convert into the nucleosides with the help of enzyme that is nucleotidases. Okay, this is extra information. Okay, so this was what, this was the digestion of the protein which is occurred in the duodenum, and it will absorbed by the villi. Okay, we'll be discussing the structure of the villi too in the soon videos. So today we have discussed about the digestion of the protein.